Keeping California's agricultural lands healthy and productive is a never-ending task which requires vast resources of knowledge and endless scientific research. In furthering such research, the University of California maintains a statewide agricultural experiment system which helps safeguard our golden harvest. Commonly known as CES, the Riverside Station is the world's largest center of subtropical horticultural research. Its scientists and technicians use an astounding variety of tools and methods in their many projects. A biotrone which regulates plant light and climate. Boxcars which go nowhere. The electron microscope which magnifies up to 50,000 times. Radioactive isotopes, dangerous but invaluable for modern research. Dealing with agricultural diseases, developing new ways to combat plant pests, and solving problems of nutritional deficiencies is only part of CES activities, however. As more and more agricultural land is cleared to make way for necessary housing, scientists and technicians at the experiment station seek ways of using all farmlands more efficiently. Whether employing standard laboratory methods or developing special field equipment, CES research is comprehensive. Now let's see how all this functions. In 1954, alfalfa growers began to suffer drastic loss from the spotted alfalfa aphid. Native to eastern Mediterranean lands, this destructive pest was multiplying explosively in America, as the red areas indicate. Chemicals were effective against the aphids at first, but it soon became apparent that chemicals alone could not economically control them. Such natural enemies as the lady beetle might help do so, however. The lady beetle thrived on aphids, as did its hungry larvae. But more help was needed, and CES entomologists looked for it in the aphid's native lands. Cooperating with United States Department of Agriculture scientists, they discovered parasite wasps, which kill the spotted aphid by laying eggs inside it. From such countries as Italy, Israel, Iran, the beneficial insects were mailed to Riverside. Here they were inspected preparatory to raising them in large numbers in the CES insectary. In due time, wasps, descended from the imported insects, were placed in centrally located colonization fields. After the aphid killers became well established in their new homes, they were harvested by a giant vacuum collector. This equipment was designed and built by CES technicians especially for the job of collecting insects in the field. Days were held and beneficial wasps were given to growers for distribution in their aphid-stricken alfalfa. Such cooperation with the farmer or rancher is an essential part of CES activities. This biological control of the spotted alfalfa aphid was and is very effective. It helped save millions of tons of hay and millions of dollars annually. Other methods of combating aphids were also used and are still being developed and used. Controlled fungus diseases were successfully employed, and today CES scientists are developing methods of treating the seeds of alfalfa so that the growing plant itself will kill insects which attack it. Such insecticides are known as systemics. This lifeless-looking powder actually is full of living microorganisms deadly to certain insects. The organisms are known as Bacillus thuringiensis. By treating plants with solutions of microbial compounds containing these insect killers, CES scientists determine how effectively they destroy pests feeding on the plants. The bacterium are harmless to vegetation, but act as a stomach poison to hungry and destructive insects. This caterpillar has just sampled some thuringiensis toxin. Now its days of destruction are over. Such living insecticides have scored major victories against plant enemies when scientists have taken them from the laboratory to the growers' fields. Fundamental research is often necessary in developing new weapons against man's old enemies. An old enemy like the common housefly, for instance. CES scientists seeking new ways to control this pest started by examining the atomic molecular structure of a vital hormone in the fly's nervous system. Assisted by an atomic modeling kit, they imitate the structure of the nerve hormone with a synthetic chemical. In theory, the chemical will disrupt the normal action of the hormone and kill the insect. From slide rules and molecules and formulas, the work progresses to the actual manufacture of the proposed fly killer. The crystals of the new insecticide are beautiful when seen under a microscope. But whether or not they are deadly is tested by treating flies which have first been anesthetized. In a few seconds, the treated fly will struggle to return to life. Then, if the scientists were right, it will die. While it seeks to control or destroy agricultural enemies, the citrus experiment station also works to maintain the good health not only of citrus, but of California's multi-million-dollar row and field crop industry. One of the most baffling problems facing science is that of the virus diseases which can quickly destroy even such healthy plants as these. Leaves from a virus-infected tomato are prepared for examination under the electron microscope. Diseased tissue is placed in an extraction solution and macerated. This disrupts tissue cells and frees the virus. Virus particles are then concentrated by whirling the solution in a centrifuge which develops a centrifugal force of 100,000 times gravity. After this, the particles will be dispersed in another solution and sprayed on metal discs for viewing. With a shadow caster, the technician coats the virus particles with metal, causing them to throw shadows for better visibility in the electron microscope. Now, using powerful electron eyes, the scientist probes ever deeper into secrets of the virus. Conquering shellbark and other virus diseases through the use of Nuceller seedlings has been a major CES contribution to citrus growers throughout the world. Many citrus varieties produce two kinds of seedlings, ordinary hybrids and Nucellers, which are usually virus-free. Production of Nucellers begins with seeds from these varieties. Sprouted in station greenhouses, they are grown to proper size for type identification. To separate Nuceller and hybrid seedlings, scientists pollinate them with the trifoliate orange. This orange relative imparts its dominant three-leaf character to the hybrid seedling only. The others are Nucellers. Budding from mature Nuceller seedlings will give the grower healthier, more productive trees. Sometimes crop troubles don't begin until after harvest. In California's packing houses, high-quality citrus fruits are carefully prepared for the traveling they must do. But it's a long way to eastern markets, and occasionally trouble develops while fruit is in transit. Such decay is caused by these penicillium mold spores. So CES scientists developed chemically treated paper to control the molds. Placed in shipping boxes, the paper gives off a gas which prevents mold from forming. The experimenters even built a stationary boxcar to make certain that the mold preventative would work under all conditions of cross-country shipping. The boxcar duplicates temperature and humidity changes, imitates varying train speeds, and records important gas concentrations. Such comprehensive treatment of research projects is one of the major reasons for the Citrus Experiment Station's many achievements. Increasing fruit storage life with 2,4-D is another CES project. Lemons treated with this plant growth regulator are stored in refrigerators and then compared with untreated fruit. 2,4-D is known to most of us as the herbicide which kills weeds without harming grains or grasses. After storage, untreated fruit from the control box has a dead black stem end, while treated fruit on the right is in first-class condition for marketing. 2,4-D sprayed on these grapefruit trees might have checked this fruit drop, as it did here. Such spraying can also mean bigger fruit size for the grower. Regular orchard spraying is also necessary for top production. The station develops new equipment and improves old methods. In the soil itself, applying proper amounts of nutrients at most efficient depths is vitally important, as soil fertilization tests at CES have shown. Increased yields and healthier crops are part of the dividends paid by combining laboratory science with practical application know-how. Because vigorous plant growth and health demands proper nutrition, CES scientists use soil columns to study plant nourishment. Soil solutions containing vital elements are drawn off for analysis, as are liquids from trees raised in special tanks. Leaves from trees grown in controlled soil experiments are also used to study soil productivity. The leaves, which are an index of nutrient status, are reduced to ashes by burning in a muffled furnace. Residual ashes are then dissolved in acid. Analysis of the liquid will help determine the amounts and kinds of fertilizers to add to the controlled soil. By learning the tree's basic mineral needs under irrigation, science helps point the way to growing better fruit. Testing and evaluating the usefulness of commercial insecticide is a regular part of CES activities. Chickens sprayed with such preparations are kept under careful observation to determine the insecticide's effectiveness. Because these insecticides possibly could leave harmful deposits in meat or egg, laboratory tests are made to determine whether or not such residues are present in significant amounts. Thanks to the station's meticulous investigations in the interest of public well-being, the consumer of California poultry products can enjoy them with complete peace of mind. In addition to being safe to eat, foodstuffs should also taste good. CES investigators use machines like this vapor phase chromatograph to measure flavor components in testing fruit quality. But no machine can replace human taste buds, so juice is tested for palatability by an informal panel of staff members. While in the nation's markets, the shopper applies the final test of quality and economy. CES scientists can't do much about the weather, but they can help orchard protecting machines guard against frost by studying orchard air conditions with instruments carried aloft in this station-designed baby blimp. When the rains come, the Southland must be prepared to use them to best advantage. Because of the growing need for efficient methods of irrigation, the Citrus Experiment Station has instituted a thorough study of soil moisture and irrigation problems. Instruments like this neutron moisture meter provide an atomic age tool for determining the amounts of water in soil. Data gathered by repeated readings will be used in water movement and recharge studies and in establishing irrigation needs for crops in various climatic zones. With some crops, such as avocados, efficient use of irrigation water is best determined with tensiometers, which measure moisture conditions at the root zones. The rancher also can use such instruments. Information gathered from readings helps him irrigate more efficiently and thus conserves our precious water supplies. But even properly watered soil can contain destructive agents. This sugar beet field has been attacked by tiny parasitic worms known as cyst nematodes, which damage plants by feeding on their roots. The white objects are cysts containing nematodes and their eggs. These are the eggs magnified several hundreds of times. In CES greenhouses, scientists examine the roots of lemon seedlings which have been infected with other species of these pests. Root tips attacked by the parasite show abnormal growths or galls. These are the sheath nematodes feeding on roots. Parasites from roots are collected in vials and prepared for microscopic examination. Magnified about 500 times, the worms are carefully studied in order to discover methods of combating their ravages. Such research pays off in better methods of applying nematicides to nematode-infested soils at effective depths and dosages. Research at CES is directly geared to the needs of modern agriculture. Rootstock to treetop, CES scientists and technicians work from the ground up in dealing with the vital problem of citrus orchard rejuvenation. Proper pruning techniques are part of rejuvenation studies because they can improve fruit quality and help lower costs. The station also works at improving rootstocks, irrigation, soil management, and development of new fruit types. Efficient cooperation by all departments is necessary in such a comprehensive program. Scientific teamwork, in fact, is as much a part of the station as is its world-famed collection of citrus varieties. One of the worst problems facing Southern California today is smog. It occurs in rural as well as metropolitan areas. An increasing liability to the Southland's economy, smog is caused principally by combustion byproducts. It is a physical as well as an economic irritant. In special smog chambers at Riverside, volunteers show the effects of artificially produced smog. A major concern is the effect on agriculture. To create smog for research, hydrocarbons and oxides of nitrogen in the yellow tanks are fed into a photochemical reactor which duplicates the effect of sunlight on smog compounds. The resultant smog is shown here, solidified at minus 112 degrees Fahrenheit. Separated into pure compounds, the smog is dispersed into plant fumigation chambers at concentrations which might actually be found in the atmosphere. Damaged plants are studied in relation to known pollution factors. Wherever practical, methods of protecting plant life are devised. Smog components produced by automobiles are collected at the source. Researchers have already cleared the way for new protective measures against this modern problem of smog, while at the same time, work continues on the many other problems involved in maintaining a healthy agriculture. But even more progress is in store for all of us, as scientists at the Citrus Experiment Station and scientists everywhere worked tirelessly to make the good earth more bountiful and more pleasing to behold.